Welcome to the online platform of Sanjivani Mahavidyalaya Chapoli, affiliated to Swami Ramananda Tirth Marathwada University Nanded. Here is Department of Botany, semester 4th and semester 6th, paper 9th and paper 14, plant metabolism, biochemistry and biotechnology for B.Sc, second year and third year student. We study photosynthesis, and in previous lecture you have seen absorption spectrum, action spectrum, quantum requirement and quantum yield, photosynthetic unit, that is quantazome, photoluminance, photon, quantum, etc. And in this lecture, here we discuss immersion, red drop effect and enhancement effect. R. Emerson and Lewis in 1943, while determining the quantum yield of photosynthesis in chlorella by using monochromatic light of different wavelengths, and he noticed a sharp decrease in quantum yield at wavelength greater than 680 nanometer. Emerson and Lewis, while determining the quantum yield of chlorella by using the wavelength greater than 680, and he found that there is decrease in the quantum yield. This decrease in quantum yield took place in far red part of the spectrum. That is, the curve shows quantum yield drops dramatically in the region above 680 nanometer. That is the red region. This decline in photosynthesis is called red drop effect or Emerson first experiment. Later on, in 1957, Emerson and his co-workers found that inefficient far red light in chlorella beyond 680 nanometer could be made fully efficient if supplemented with light of short wavelength. Emerson and his co-workers in 1957 in his second experiment found that inefficient red light in chlorella beyond 680 could be made efficient or fully efficient when you supply the short wavelength of a light. The quantum yield from the two combined beams was found to be greater than the effect of both beams when used separately. This enhancement of photosynthesis is called Emerson enhancement effect. Emerson and his co-workers found that when the red light above 680 nanometer, when supplemented to the chlorella plant, there is a sharp decrease in quantum yield. And in his second experiment, he found that when shorter wavelength of light with far red light 680 above combinedly supplemented to the chlorella plant, there is a enhancement of quantum yield, or that is the enhancement of photosynthesis found. First effect is known as Emerson red dwarf effect, and second effect is known as Emerson enhancement effect. Light trapping centers, that is photosynthesis 1 and photosynthesis 2. The discovery of red dwarf effect and Emerson enhancement effect concluded a new concept about The role played by chlorophyll A and accessory pigments in photosynthesis. After the discovery of red dwarf effect and enhancement effect, the concept about the role played by chlorophyll A and its accessory pigments in photosynthesis, that photosynthesis involves two distinct photochemical processes. The photosynthesis involved two distinct photochemical processes. These processes are associated with two groups of photosynthetic pigments called pigment system or photosystem first and photosystem 2. Each pigment system consists of central core of complex and light harvesting complex. Each pigment, that is a group of pigment, consists central core of complex and light harvesting complex, that is LHC, light harvesting complex, comprises antenna pigment associated with proteins, that is antenna complex. Their main function is to harvest light energy and transfer it to their respective reaction center. Photosystem 1 and photosystem 2. That photosynthesis is associated with two reaction center, photosystem 1 and photosystem 2. Each reaction center in photosynthesis contains a group of pigments. In that group of pigments, a central core is present, that is known as light harvesting complex, and with that light harvesting complex, the associated pigments are also present. That associated pigments act as antenna pigments. The role of antenna pigment in their group of pigment system is that antenna pigment harvest light and supply to the reaction center. Wavelength of light shorter than 680 nanometer affect both the pigment system, while the wavelength longer than 680 nanometer affect only the pigment system 1. Listen that carefully. The wavelength of light shorter than 680 nanometer affect both the pigment system, while the wavelength longer than 680 nanometer affect only photosystem 1. That photosystem 1 is found in thylakoid membrane and stroma lamellae. It contains pigments chlorophyll A, that is 660 nanometer, chlorophyll A 670 nanometer, chlorophyll A 680 nanometer, chlorophyll A 690, chlorophyll A 700. Chlorophyll A 700 or P700 is the reaction center of photosystem 1. P700 is the reaction center of photosystem 1, and photosystem 2 is found in thylakoid membrane, and it contains chlorophyll pigment 650, chlorophyll A 660, chlorophyll A 670, chlorophyll A 678, chlorophyll A 680 and 690, and phycobilins. That P680 or 690 is the reaction center of photosystem 2. Chlorophyll pigment A is found more in photosystem 1 than the photosystem 2. Carotenoids are present in both photosystem. Photosystem 1 is associated with both cyclic and non-cyclic photophosphorylation, and photosystem 2 is associated only in non-cyclic photophosphorylation. Both the pigment system are believed to be interconnected by a third integral protein complex. Both the photosystem, that is photosystem 1 and photosystem 2, are interconnected by a protein complex that is called cytochrome BF complex. The other intermediate component of electron transport chain, that is PQ, the plastoquinone, and PC, that is plastocyanine, act as a mobile electron carriers between two pigment systems. Photosystem 1 is active in both red and far red light, and photosystem 2 is inactive in far red light. Evidence in support of two phases of photosynthesis. First, physical separation of chloroplast into grana and stroma fraction. It is now possible to separate grana and stroma fractions of chloroplast. If light is given to grana fraction in the presence of suitable hydrogen acceptor and is complete absence of carbon dioxide, then assimilatory power ATP and NADPH2 are produced. If these assimilatory powers are given to stroma fraction in presence of carbon dioxide in absence of light, then carbohydrate is synthesized. Second evidence, temperature coefficient, that is Q10. Temperature coefficient is the ratio of rate of reaction at a given temperature, and a temperature is 10 degrees Celsius lower. The temperature coefficient value, that is Q10 value of photosynthesis, is found to be 2 or 3, that is for dark reaction. And when photosynthesis is fast but Q10 is 1 for light reaction, when photosynthesis is slow. Third evidence, the intermittent light. The Warburg observed that when intermittent light, that flashes, intermittent means flashes of light of about 116 seconds were given to green alga, the photosynthetic yield per second was higher as compared to the continuous supply of same intensity of light. This confirms that one phase of photosynthesis is independent of light. The evidence from carbon dioxide in dark reaction or in dark. It comes from tracer techniques by the use of heavy carbon in carbon dioxide, that is C14. The leaves which were first exposed to light have been found to reduce carbon dioxide in dark. It indicates that carbon dioxide is reduced to carbohydrates in dark, and it is purely a biochemical phase. Here we stop, and in next lecture we see the light reaction, that is the photochemical phase. Thank you.